Hello. Hello, everyone. Thank you for joining us today. We're going to wait a few seconds so more people can join and log in, and then we'll start the webinar. So please wait a few seconds. Okay, so I think we can start now. Well, hello, and thank you for attending our Early Career Members Network Live webinar series. My name is Daniela Ramirez. I'm a postdoc researcher from UNCuyo University and CONICET fellow from Argentina. I'm also an EMN committee member, and today myself, along with Juan José Oropeza Valdez, who is a postdoc researcher from the Laboratorio de Biología de Sistemas in the Universidad Nacional Autónoma de México, and also an EMN member. We will be both co-hosting the 20th session of the EMN live webinar series from the Metabolomics Society. We hope you enjoyed this session, but before introducing today's speaker, we wanted to remind you this session is going to be recorded. Also, if you haven't done it yet, we encourage you to join our social media networks via X or Twitter, or following our LinkedIn page for updates or for featured content. You can also send us an email to make suggestions or ask for further information. And we're recently in Blue Sky as well, so you can find us there too. In this opportunity, we have the pleasure of presenting Dr. Farhana Pinu. Her research work is entitled Flavoromics, a New Frontier in Food and Horticultural Science. We truly appreciate Dr. Pinu's presence today, and we believe it's a really interesting topic, and we hope you enjoyed this session. Now I'm going to let Juan introduce her properly, and then Dr. Pinu can start sharing her screen.
Thank you all for joining us today. It is my pleasure to introduce today's webinar speaker, Dr. Farhana. Dr. Farhana is a science group leader of Biological Chemistry and Bioactives Group from the New Zealand Institute for Plant and Food Research, also known as PFR, where she oversees 4 different teams that specialize in metabolomics, analytical, and natural products chemistry. After completing her PhD from the University of Auckland, New Zealand, she joined PFR to work on a program to develop fermentation technologies to produce lower alcohol wines using data-driven approaches. Over the decade, she has contributed to some of the pioneering work involving the application of targeted and untargeted metabolomics in grape and wine research. Since 2021, she has started expanding her research on other food and horticultural products. Therefore, her current projects focus on the development and optimization of metabolomics, lipidomics, flavoromics, and imaging approaches, mainly using mass spectrometry to understand plant systems, identify new flavor traits in horticultural product crops, and also to study microbial metabolism linking to fermentation outcomes. Also, Dr. Farhana was the president of Australian and New Zealand Metabolomics Society from 2020 to 2023. Ladies and gentlemen, without further ado, please welcome Dr. Farhana, who will be speaking on Flavoromics, a new frontier in food and horticultural science. Please, Dr. Farhana.
Thank you, Juan. I'm just going to take some time to share my slides. Just bear with me. Okay. So can you see it?
Yes.
All right. Thank you so much for inviting me. I know that many of you joined from different parts of the world. Although you're saying it's May 14th, but for us it's May 15th. So we are in Wednesday, not Tuesday. Oh, Thursday today, sorry. You are in Wednesday, we are in Thursday. So I chose this topic and I'm kind of going to do it more like a workshop style rather than going through too much of the data. And you will be hearing from me of some of the things that Metabolomics Society or Metabolomics community might not have done before, but this is why maybe it's interesting. So the Flavoromics is actually a pretty much new area for me as well. I'm still learning. So I do have my colleague, Emma Sherman, who is the true specialist in this approach here. So I might call in her if some of the questions you are asking me, if I cannot answer them. So here is the kind of outline that I have decided for this presentation. So I'm going to quickly introduce you to plant and food so that you have better idea why we actually started doing Flavoromics. And then that will follow on, of course, explanation of Flavoromics, what and why we're doing it. How does the workflow look like? What do we need to consider? And after that, I will give you some examples from published research that how this field is developing and some of my favorite papers, of course. And at the end, actually half of the presentation will be 2 case studies that we have. One we finished and another we are still doing. And here, as you can see, this is a pear orchard in one of our plant and food, side. And this is our sensory colleagues working really hard to run the sensory panels. You'll see some photos here and there so that you have better understanding of what we do. So introducing plant and food, I just want to mention one thing that often we get omitted from the map. So if you look at this whole world map, this is Australia, and we are just that little dot here, just right here. So we do exist in the map. And plant and food in this little country, we have, all over the both North and South Island. So we have around 14 sites, as far as I recall, within the country, and we also have presence in Australia, in Queensland and South Australia, and we also have a business development manager in Oregon. So we are, for a small country and small population, we are around 1,000 staff members, around more than 600 scientists working for the organization, and we call ourselves a Crown Research Institute. So we are not a university. So we kind of do both fundamental and applied research, but our main aim is to help New Zealand's primary industries. So New Zealand is known for, premium quality product. We export a lot of fruit and sometimes vegetables as well. So these are the main sectors that we have. So our work is just not only on the food, typical food, fruit, or food ingredient space. We also develop technologies, and we also grow fish as well, technologies related to aquaculture. We are contributing towards developing new cultivars. This is one of the key work we do here. So, for example, our cultivars are now growing in more than 30 countries. Some of the examples you may have seen here. You most probably know the green kiwi fruit, and this is the gold one, developed quite a while ago. And the new introduction is this red kiwi fruit. It's getting actually really good feedback from the consumers. And this is kiwi berry. So little berries that bite size. You don't need to scoop or peel. You can just pop in your mouth just like any other berries. So raspberry here, this is also the variety developed in plant and food. In addition to these fruits, we also grow vegetables like potatoes. And I heard recently that we actually export quite a lot of onion seed, which I did not know before. So this is just showing the, how much we are actually contributing to in this sector. So to develop any new cultivar, it's really, really important to understand what consumer likes and dislikes. So consumer liked unique flavor traits, identifying them, characterizing them, and advising our breeder friends is quite important. And this is why the flavor comes. This was developed by Copilot. It's really intense, as you can see, and flavor is actually intense. It's not that simple. And you'll be hearing from me, I'm talking about it's complex. It is complex. So it's a complex sensory experience that combines taste, aroma, and other sensory inputs. So flavor actually arises from combination of chemical stimuli and other human aspects, that way we perceive different flavor. I'm going to show you now my favorite quote. So this is by Shepherd. It's like flavor is not actually in the food. Rather, it is created by the brain through the interaction of stimuli with multiple sensory, motor, and central nervous system. So if you ask a psychologist or a sensory scientist, they will be calling flavor is more like psychological or cultural. But as a chemist or a metabolomics person, I'm going to also not back down. I'll say that food composition has a big role that actually can influence those motor and central behavioral systems. So what is flavor and flavor perception? Again, it's not simple. As you can see in this photo here, right? For me, the way I see it, when we talk about flavor and if we like a food or not, or a fruit, it starts all in the visual, right? In the eyes. When we see different colors, we feel attracted to it. When we see the color is not good, we often feel, no, I'm not going to eat it. I'm not going to try it, right? So we start from the eyes, right? Then when we smell something, then we can detect different aromas orthonasally, that is through the nose, like in the nose, and then also retronasally. When we bite or sip something, it also creates those aroma sensations in the mouth as well. The tastes are primarily detected by the receptors that are present on the tongue. So I was just doing some Googling yesterday and realized that we have thousands of taste buds in our mouth. And each taste bud has like 50 to 150 receptors. So that means like so much is involved in there. So this is why I'm saying it's not simple. So in terms of flavor perception, texture, mouthfeel also contributes significantly. And there will be a lot of other nervous systems included as well. For example, some of the mechanoreceptors, when we touch something, some of the thermoreceptors that gives a sensation of hot or cold. So this is why it is really, really complicated. And flavor perception is mediated by human physiology and psychology. But again, human physiology and psychology can be influenced by the composition of food. So that brings me to the chemistry of flavor. So this is something that has been done for a really long time, and we all know it, that if we look at the composition of any food or beverages, there are many, many flavor active compounds that directly contribute to aroma and taste. Here I'm mentioning some of those basic taste. I'm not going to explain quite a lot because we all eat food, so we know that among the basic taste, we have sweet, salty, sour, bitter, umami, and recently kokumi as well. So these all tastes contributes to the overall flavor perception. And then aroma, as you know, we have esters, terpenes, and pyrazines. They have their different type of notes that we smell. When we smell them, some of them we like, some of them we dislike. So among aroma, it's not everything is desirable, right? So we are individually speaking, we like different aromas. Some people like fruity aromas, some people like citrus aromas. So there are also a bunch of other chemical compounds or volatile compounds that we might not like. So this is why I'm talking about here, the flavor enhancer. So there are, as you know, when we analyze a food, there are thousands of compounds present there, and not everything is contributed directly to the flavor. So some of them can enhance the flavor perception that makes the fruit or beverage complex. And there are also flavor neutralizers and suppressors, so that those compounds can actually neutralize and tune down the perception of flavor. So this is why it's so complicated, and we really need to go and dig deeper to understand the interaction between taste and aroma to understand the flavor term better. So how do we measure flavor? This is not rocket science, as I was saying, that this has been done for a really long time. So again, as a chemist would say, the analytical chemistry is the best approach to characterize the flavor compounds. So we can do them in a very different way. We can use just simple bench chemistry, spectrophotometry, spectrophotometer, use them to understand what are the compounds present in the food. And also, in addition to the analytical chemistry, and you will be hearing from me more about sensory and integration side of things, that sensory studies we can conduct actually use the human being to describe the psychological and cultural aspect of flavor and its perception. Sensory studies can be done in many different ways. So among them, mainly used approaches are using expert panel. Who are the expert panels? They can be the winemakers, they can be the bakers, and our breeders as well, because they are always tasting what they're making. So that's make them experts. So we can use them and give them something to try and tell us what they perceive from there. You can also use trained panel. So you train a specific group of people and so that they have better understanding how to describe the flavor. And you can go directly to the consumer, because at the end, they are the main people going to buy it. So to getting idea from the consumers will provide an overall idea about what they like or dislike. And then the integration part, we can combine both these approaches together, and we can try to isolate, characterize the molecules that are related to sensory perception. We can do some addition omission tests to understand what needs to be there to create that particular flavor. And we can also use GC olfactory if we kind of pinpoint with the other analytical chemistry approach, we can then use that as well. So traditional flavor chemistry, if we compare with flavoromics, traditional flavor chemistry is mainly hypothesis driven. You already have a hypothesis, you design an experiment, you do the data acquisition, which is often targeted. For example, I still remember when I started my PhD, if we could actually analyze 50 compounds, that was a large set of data, right? But that was very targeted type of approach. Now we can do more than that. So traditional flavor chemistry mainly rely on validating a hypothesis. On the other hand, flavoromics is a data-driven approach. So we do have a problem, research question, we might not have a hypothesis from there. So we are using the data-driven approach to generate hypotheses. Depending on the research question or problem, we design experiment, we do data acquisition, which are usually non-targeted, but sometimes we do take semi-targeted approach as well, because as you know, non-targeted approaches can be quite complicated, especially data mining side of story. It's quite complicated. We still deal with a lot of unknowns. So sometimes when we don't know something, that can be quite tricky to deal with. So that means that in flavoromics or any data-driven approaches, data interpretation is really, really important. And then we can generate a lot of hypotheses. Those then we can use some flavor chemistry approach, hypothesis-driven approach to prove or disprove that particular hypothesis. So now flavoromics, again, Emma always tells that we are not the one introduced the term, because these days it's really trendy, right? So adding omics after everything. So flavoromics is actually a real field, and it's a sub-area of metabolomics. So we are not competing with any other omics technologies. We are actually a branch of metabolomics. And this term was initially introduced by a renowned professor from Minnesota in 2008. But the first publication actually did not come out till 2011. I'll talk about this a little bit. So the way the professor described it, it was a non-targeted, data-driven approach aiming to, to correlate the chemical composition of foods with their sensory properties. So that means that Flavoromics directly contributes to a better understanding of consumer perception and preference. This is why Flavoromics, because we are integrating chemistry with sensory science, can be quite powerful tool. This is just, I took it out from the case I had to write for Flavoromics team formation back in 2022. This is our side of story, like why we are using Flavoromics in plant and food. So because we want to help our breeders to identify novel and unique flavor traits so they can actually enhance their breeding pipelines. We also help a lot of our industry partners and also scientists to develop knowledge and technologies about, about the food so that they can develop better quality products with by understanding the desirable and undesirable sensory characteristics, what people like or dislike. And also using Flavoromics, you can actually generate recommendations that can help developing new food and horticultural products based on consumer preferences. It's just not like what I want, what people want. And we also use this, like the knowledge we are generating here, we can also use this to raise consumer awareness about food and horticultural product based on science, just not on just, the social media post. It's just we are using real science to describe or understand the food products that consumer will benefit from. So key points to keep in mind. This is very similar to any omics technologies. We have to design the experiment properly. If we don't design the experiment properly, there is no meaning to collect so much data. And then at the end, the data means nothing, or we misinterpret that and come up with wrong hypotheses. That is not going to work. So experimental design is the key, and also quality control. We have to be very, very careful because we are dealing with untargeted data mainly. So we need to make sure that we are doing proper quality control, using the proper quality control measurements. And also appropriate statistical and data analytics. This is really, really important as well. As I said, if we misinterpret the data, that means it's not going to benefit the science in any way. So we have to be very careful here. So if we fail either of those steps, Flavoromics workflow will not work. So we have to think carefully and consult experts before we do any experiments. So this is a very multidisciplinary approach. So we have to talk with a lot of other experts from other fields. Especially, I would specifically mention about the sensory experts, because they know the psychological or cultural aspect of the flavor perception. So we have to work with them very closely. Now the overview of the Flavoromics workflow. I'm not going to spend too much time on this part because you all know it. You are all specialists. You know how to do metabolomics. So I'm going to say that, okay, these are the kind of examples of samples we use in plant and food, but other people use it for other products as well. So this is like metabolomics workflow as usual, that we are generating the food metabolomics type of data, but the main thing is here, the sensory study, okay? And really important thing is combining the chemistry and sensory data to develop predictive models and hypotheses and performing as usual the characterization, isolation, and other tests. And at the end, we get the biochemical basis of flavor perception that actually can give us a lot of idea about the psychological and physiological aspects from the human as well. Looking at the publications, so far since 2021, there was 132 publications. That was just a Web of Science search, so that might be others as well. So the growth of Flavoromics is pretty slower compared to other omics approaches, I would say. Very it started to grow, I would say, from 2022. 2025 is not over, so do not take it as a slump. It's just the year is not over. So I'm hoping that by this year, this will go even far. But one of the key trend, or I am particularly concerned, the way we actually claim things. There are many of these publications, they are not actually flavoromics, they are doing untargeted volatile profiling and claiming it as flavoromics. So yeah, so this is why if I'm one of the reviewer, I would like to see that where is the human aspect of the human flavor perception as data sets are. But again, you know, because some name is it sounds quite cool, people like to use them. So I just want us to be really clear that for flavoromics, we really need to do both chemistry and the sensory. Otherwise, we should not claim it's flavoromics. So this approach has been used not only for horticultural products. As I mentioned before, I have seen papers on dairy products, even poultry, meat, and so on as well. So it's growing field, and I think once we can show that what this approach can do, it will keep growing. And now I'll just show you a couple of papers, really, really good ones, and also the first ones as well. So mainly you will see these publications in Journal of Agriculture and Food Chemistry and Food Chemistry. So they are kind of publishing quite a lot on flavoromics as well. So one of the study was done on understanding the freshness in oranges and tangerines. The coffee quality, I really, really like this study. You know, it's really nice. It's been used for hops cultivar screening as well. And last two from us, so I'm biased, as you know. To understand the wine quality. And my direct involvement started in flavoromics with this paper, which I'm going to talk very soon. So that brings me to the first case study that we used flavoromics to investigate the fate of aroma compounds in the mouth. So as I was telling you, Emma Sherman, she's one of the team leader within my group. She's leading the flavoromics team. She did her PhD, started back in 2011, I think, as far as I recall. So her PhD was applying flavoromics to understand Pinot Noir wine quality. So when Professor Maria Del Pio Bayon and Maria Perez Jimenez, they contacted me to collaborate in a project back in 2019. I thought, okay, I'll convince Emma because I'm not the specialist. I know how to do metabolomics. I'll ask Emma if she will join me to help Maria to finish her study. So Maria came from Spain to New Zealand and brought a lot of samples with her. Some of them were lost in our really tight border security. If you have not been in New Zealand, our border security is really, really strict. So we lost something, but we also gained quite a lot as well. So we wanted to use like flavoromics approach to understand wine aroma perception. As you know, we are all different. So, and individual variation can be dependent on age, gender, other factors in the mouth, even your mouth, like saliva microbiome or composition can play a big role. So we do need to understand how wine and saliva metabolites are interacting with each other. And that actually plays significant role the way we actually perceive a wine. So aroma perception when we're drinking wine, I'm just going to spend a little bit time because some of you might not be familiar with it. When we look at the color of the wine and we smell it, so this is the pre-ingestion type of phase. You know, we are actually kind of our nasal olfactory receptors are getting activated, and we ingest something. So we are feeling the aroma sensation here in the nose. And the phase 2 is the ingestion after we drink the wine, and then it gets mixed up with our saliva, and our taste receptors also get activated, and then we feel the aroma retrodensely that is in the mouth. And then post ingestion as well, there will be a lot of volatile compounds I think non-volatiles as well. They can stick to our oral mucosa, and we can actually further feel a lot of other aroma compounds as well. And some of them stays there. So that is all called aroma persistence. So to design this study, this was done in Madrid. So they have their team is very specialized in wine oral processing. So what happens when we are drinking specific drinks, not only wine, they work with other stuff as well. So it's called speed of odorant measurement. So as you can see, you have a panel and you kind of first give them some water to rinse the mouth. So this is the control. So this is basically water plus saliva. And then after 15 minutes, the first wine rinse. Okay, so you take the sip of the wine, you don't drink it, you just spit it out after 30 seconds. So that's the expectorated wine. And there is second rinse, and there is a third rinse. And then they combine this together so that they have like a little bit of better sample matrices that has both wine and saliva. So they used a red wine from Rioja. And then Maria brought the samples to Auckland. And as you can see, the participants' information, there were only 32, but there were a lot of samples. So they divided the participants based on their age. So young, 18 to 35, and senior, 55 to 72. You might disagree, but this is how they divided the age groups. And these were the samples I already kind of explained to you. And we did some untargeted volatile profiling. And we also had a lot of pooled quality control samples. We combined all the samples together, and we used them for data quality checking and also the instrument variation, because we could not run those 300 samples in one day. So batch to batch variation, and also sometimes we use them for normalizing untargeted data as well. Untargeted volatile data can be quite messy, so it requires a really long time to do the data mining and cleaning. So this is basically the overview of the data. As you can see here, we have the quality control, quality control. Wine itself, and also wine expectorated. As you can see, there are the saliva and water here and here as well, and they are completely different than the other samples. Like this is the quality control samples here, and these are the wine expectorate, and this little blue dot is here the wine. We had initially had a lot of features to deal with, over 1,700 across all the samples. But Emma went through a lot of cleaning steps, and at the end, we ended up with 248 features, which we used for the statistical analysis. And after statistical analysis, we were only able to putatively identify 30% of them. So this is one of the bottleneck. We still have not everything we can identify. There are lots of unknowns in the samples. So what happens to wine volatiles in the mouth during wine consumption? So in those samples, there are a lot of different types of, chemical classes. So we all confirmed the previously known observations from Maria's team, so those are nothing surprising, already known. But we also, this data provided us something new, some more new indication, hypotheses that might be happening during the wine drinking. So for example, the metabolism carboxylic acids during the wine oral processing, also esterification. And we also saw some production of new volatiles in novel generation of volatiles from non-volatile precursors. So these are some of the examples. So we also saw that depending on our oral hygiene, and there are some compounds related to the, the toothpaste or mouthwash we use. Although they did a lot of rinsing before doing the, collecting the samples, but there's still some leftovers left there. So there was quite a lot of interesting information that we tried to look at, and we generated some new insights that were not known before. So we published the study back in 2023. Now my question is, this data was quite interesting for me, and does the age matter? So although we all want to think we are all young in our heart, but age does matter. And the changes pattern that we saw within the samples are dependent on consumer age group. For example, relative abundances of most of these compounds were higher in seniors than young consumers. And Maria's group actually did a lot of work, and they actually showed that saliva flow, saliva composition, saliva microbiome is quite different between different age groups. But in this study, we found that the gender, when we combine all the data together, we almost don't see any gender-related difference. But when we divide them, look at the interactions between age and gender, and we see that gender-related differences were more pronounced in young consumers than seniors. So the young people samples are more variable than the senior people samples. That means that, yes, age matters, and the way we perceive different wine could be related to age. For example, I often hear that senior people would like a really heavy red wine. On the other hand, young people might like something a little bit lighter, not that heavy. For example, a rosé or sparkling sort of wine. So depending on this, so what we got out of from this study, that means that we can actually make some winemaking adaptation changes to improve the mouth aroma release. So that means we can develop some new additives that will improve the aroma resistance and reduce the off flavors. And also, depending likings and dislikings, we are generating information to understand the consumer behavior better. And with this information, can we actually group consumer based on how they perceive or how the aroma compounds are evaluating in their mouth? And this is just like my wish list. I would like to, go for more systems biology type of approach and get a lot of data, but most of the time, we don't have enough money to do everything. But, if we could do it properly, combine a lot of data-driven research, we could actually develop data integration and modeling approaches to group the consumers. And depending on what they like, winemakers can go and make it. So it's just not only for winemakers. This is just an examples. It can be applicable to other product as well. So this is my case study number 1. That was a processed food, fermented food, and a liquid product. But now I'm just going to give you another example of main thing that we work within plant and food is the fruit horticultural product. Understanding pear flavor and flavor perception. In New Zealand, we grow 6 to 7 varieties, mainly European pear. If you know about pear, that there are 2 main groups. One is European pear and another is Asian pear. In New Zealand, we mainly grow European pear, but there are some Asian pears grown here too. So as an institute, we have developed this little beauty. It's a cross between Asian and European pear, and it has distinct flavor and texture, and people are actually receiving it pretty well. One of the key things is that in pear, when I was looking at the literature and not much information in the flavor space is available for European pear. There are a lot of studies coming out from especially China, South Korea, and Japan on pear, but not much on the European pear. There are studies, especially on the, we work on pear, but in like post-harvest disorder space, coughing, on other things. We did not have in the flavor aspect, we did not have any project until this man, he's spending just a year with us. He's from Chinese Academy of Agricultural Sciences. He came to New Zealand last year and he's a pear breeder. So we wanted to design a project for him where he can learn Flavoromics. This is a photo just from after our Christmas party. We're called we're all quite a little bit of tipsy here. And we designed this project mainly for Xinguang. Here is the kind of project, how that looked like. There is no blue pair. It's just for me to just give you a visualization, the different varieties. And you're noticing that I am not putting the full name. As an organization, we do have a little bit of confidentiality that we need to maintain until we publish or we get approval from our industry. We don't say the name, although these varieties we directly purchased from the fruit store. They are not sensitive commercially, but I'm just, because we haven't published this study yet, so I'm just going to omit the names. I'm just giving them random code name here. So what we did, we bought the fruits from the store, and then it went to a little bit of post-harvest intervention to make them similar type of, like how long they're staying in the storage, and also eating ripe type of situation. My post-harvest friends can tell that better. And then what we did, we kind of did sensory consumer panel. 63, they are all planted food stuff, 30-minute session. So this is just because it was a small study. We did not, and also we did not have funding to go and go for like external sensory panels. So we just did it in-house, and we got really, really good response. And our colleagues were really happy. They are not only scientists, there are other colleagues as well. So it was a good mixture. They're the real representation of the consumers as well. So that was 30-minute session. And we kind of gathered data in terms of overall liking, disliking. And there is a technique called check all that apply, developed in, not developed, but widely used in plant and food. We collected ortho-retronasal aroma description data, also emotional responses, conceptual responses. And the sensory data collection was using CompuSense cloud software. After we did the sensory, we took the samples to our lab, and we separated the skin, flesh, and also created our quality control samples. We did the volatile profiling. So today I'm going to show you the combination of volatile profiling and the sensory data. We are still doing this part. And we'll explain a little bit more what we have done. Just to show you, so the post-harvest data, what we measured, we did the fruit firmness, total soluble solids, textural acidity, also dry matter, and also the ethylene production, dynamic ethylene production over the time as well. So those data I'm not showing here, but this is just to give an example. The different pear varieties, their firmness are different. Soluble sugar contents, if not too much different, and the textural acidity, not too much different, but they are different. And this is normal kind of data. And this is just to show the sensory crew behind the scene. I was there as well. I was the photographer. I was hands-on those 2 days. But they did really work hard, and another bunch of people were working in the lab, chemistry lab. So because we have to prepare the samples pretty quickly, so we had to be all the hands on the day. So this is the snapshot of how the volatile profiles of New Zealand pear looks like. I think there are around 175 aroma compounds that we are showing, and they have different type of aroma notes. I'm just showing a little bit of examples. There are compounds related to vanilla flavor, coconut flavor, related to melon notes. Sometimes the off-putting flavor could be the vinegary, acetic acid type of note, and also grassy kind of notes as well. So the main pear compound is called pear ester, ethyl decadenoate. Dienoid. So this is that compound. As you can see, they are quite different in the samples. When we are analyzing them and we put all the samples together here, and we can see the volatile profiles of European pears were quite similar to each other. This is why they are all clustering in this area. Here is a quality control samples, really nice tight cluster that shows that our data quality was quite good. And this is the variety that was developed in plant and food. It's separated from the rest of them because it's a hybrid between European and Asian pear. So its aroma profile was completely different. And this biplots showing what kind of compounds are separating these 5 different varieties. And also just we looked at the flesh and skin separately. Although again, the European pears are here and the plant and food, the hybrid varieties here. But as you can see, the clustering patterns are a little bit different between the flesh and skin, right? In terms of flesh, these 3 varieties are very similar. And this one, this pH one, is a little bit of different. On the other hand, when we look at the skin, this one is different from these 3. So, the variation, like when we look at the volatile compounds, now you can decide, like, I know that in many countries we peel the skin before eating the pear. So you might be missing some of the interesting aroma compounds if you are getting rid of the skin. So the flesh and skin volatile compounds could be a little bit different. And here, just to show that relative concentration of volatile compounds were actually higher in skin than flesh. But having said that, when we are eating a pear, we mainly, when we take a bite, the skin is very small amount compared to flesh, right? But because of this, when we did the volatile compound, we use the same amount of skin and flesh. So this is why it could be like that. But when you're eating the pear, that the concentration of volatile compounds might be similar, but because of the sample amount was higher, the concentration was higher as well. Now I'm going to introduce something that you might not have seen that much. This is the overall liking, disliking data from the sensory study. The clustering pattern was somewhat similar to the volatile profile data we saw. Like here, the PB is here, and other varieties are clustering this side. And this was one of the mostly liked variety as well. I'm not saying that because we developed it, but this is actually a unique pear. So this is the normal sensory data, how it looks like. So you have lots of aroma notes. For example, here you can see this this pH that was like nutty flavor was quite prominent, and this green grassy type of flavor. On the other hand, this BB, AL, WN, they were dominated by this type of flavor and aroma notes. And here the PB had like Asian pear flavor or tropical aroma, melon aroma, sweet aroma. Those were the main characteristics that were making this one different. This is my favorite type of things. So we usually just go for conceptual flavor perception responses like this, but I was really, really keen and kind of, my team was excited too. We generated some emotional responses. For example, when you are eating a fruit or drinking something, it makes you satisfied, dissatisfied. It gives you the feeling of complex, or it can make you excited, feel inspired, enthusiastic, refreshing, premium, indulgent, interesting, comforting, happy, satisfied. So you can see the different pear varieties. There are different type of taglines, right? So this is really, really interesting, and I am like really, really hopeful that we could do something to understand these emotional responses better. The integration part. Now, Emma spent quite a lot of time doing some partial least square regression models that provides a predictive linear relationship between variables. But when I was looking at the data with Emma, and we realized that some of the lines were not linear, it could be quite curvy as well. So I'm just showing you two examples because we don't have quite a lot of time. So this is one of the aroma note where the tropical aroma from the flesh. And the model was decent. We won't say that it's a really, really good model, but it was decent model. And all the information that's here, you can see. So if we look at this tropical flavor and the correlation plot, this compound is positively associated with tropical flavor. But on the other hand, here are too many compounds, right? But these compounds are like green, green notes type of compounds, a lot of acid and ketone alcohol, some of the alcohols as well. They are negatively associated with this tropical flavor. So this is how to explain that. So that means if we want to increase the tropical flavor, we need to reduce the concentration of these and increase the concentration of this compound. So this is all about the balance, and also again all about what the consumer wants. So another example, this is a little bit of not considered as a good or desirable character in fruit, green grassy aroma. But this was an excellent model, as you can see from the data here, pretty linear. So this one again, this is green grassy, and here that other compound I was showing that was positively associated with the tropical flavor is now negatively associated with green grassy aroma. And the same compound, like here, same compound that were negatively associated with the tropical aroma are positively associated with green grassy aroma. That means that, green grassy aroma and tropical aroma, we can now actually suggest to our breeder friends that, okay, if you want to reduce the green grassy aroma, increase the concentration of that ester and, reduce the concentration of this. So we can give them some recommendation based on this type of data. We are still looking at the data. It's not finished yet. And we are also working on a publication right now. So I'm not able to give you any more detail or too much explanation, but I'm happy to answer some of the questions, maintaining the confidentiality. Where I want to go, or we want to go, for example, this is the emotional responses plot, right? Like I'm hoping that to see this. That means, for example, all those emotional responses we see here, can we associate them with specific metabolites? It might not be one, it might be multiple compounds, but to identify that, what makes people happy, what makes people excited, what makes people satisfied, what makes people to feel at ease. This type of approach is just not for breeding. It would be quite good for understanding the other food products as well. Or it's not only food products. It can be maybe we can, I talk about, can we make a bottle of happiness, or can we do something that will make us excited? So this could be quite interesting. It's not going to be an easy road because individually we are all different. So we are not all same. Something you like, I may not like, and something I like, you may not like. So we need to take that into consideration. And we need a lot of data to gather before we can get there. But we are taking some small baby step just to, get our understanding improved in this space. But watch this space. Hopefully we'll be able to share a little bit more story around what we are finding. No, we are not only working on wine and pear. The current projects we have with the Flavoromics team and with my wider group, we are applying Flavoromics for blueberry, kiwi berry, and we are also working on some hops. And one of our big tasks is to screen kiwi fruit for the novel traits with our internal collaborators and industry partners. And we also do like validation of flavor essences. There are 1,000 compounds in food. They're not all important. sometimes 3 or 4 compound can give you the specific, for example, it tastes like gold kiwi fruit, or it tastes like apple. So we can do that as well, and how the color and other, texture, mouthfeel influence the flavor perception. This is one of the key things we are interested to. Now, at the end of my presentation, I would like to give a big thanks to the team. The wine oral processing study we have done with Thiel in Spain, with Professor Maria Angeles del Pollo Baun and Maria Perez Jimenez. And my big thanks to my colleagues from Consumer and Product Insights team, David, Petra, and Bev, especially David. He was so hardworking and did a lot of those figures for the sensory as well and run the panel. Paul helped with our post-harvest measurements. And of course, the Flavoromics team, Emma, Zoe, and Mindy, they did a lot of work. And of course, the pair study was designed for Xinguang. So I cannot ignore him, right? I could not find a good photo for him other than this, but he has done a really good job with us for last 1 year. In addition to this, the flavor research within my group is not only the Flavoromics team. We widely collaborated, although I could not show you any examples. Hopefully in future, I'll be able to show that. So I have 4 teams around the country in 5 different sites, and they do different kind of analysis. They have access to different kind of instrumentations. We also collaborate with universities if we don't have certain instrumentations. And so here are some names, especially we work very closely with Heika's team. They specialize in metabolomics, lipidomics, with Jenin's team as well. They do targeted proteomics, neurotransmitters, and a lot of food biomarker. Nigel Joyce, he's our main guy. He's doing a lot of omics integration using untargeted approaches. And Nigel Perry's team, we have a natural products chemistry team. So if we don't know something, we can go that, isolate and characterize with their team as well. So I would like to thank all of them because actually hard work is done by them. Thank you. If you have any question, you can ask me now. I'm going to stop the presentation so that I can see you.
Thank you so much, Dr. Pinu. It was a really enriching and a really interesting presentation. I think it was really interesting to hear how you explore the flavor chemistry, how you combine sensory analysis and flavoromics, also make us think other ways, other possibilities to innovate in the food production, in the beverages production. I think it's really, really interesting. So we have a few questions. Yeah, we got one question that says, is there significant variance for taste and olfactory receptors between each individual? And how does this influence guidance of optimizing for certain flavors goals for widest breadth of population?
Interesting question. Yes, definitely. As I was telling, that individual to individual, we are different. It's not only that, for example, if you remember during COVID, people who got COVID, they lost their taste and smell sensation, right? So the different disease condition also can impact how our taste and olfactory receptors can be different. And we can use actually food to, for example, some of the food component can be useful to, deal with that type of situation. The reason I'm saying that is really complicated because the individual to individual variation is such a high. This is why I'm talking about grouping. For example, ethnically speaking, the different country-wise, we are used to a different type of food. We, for example, like we grow up using maybe eating kimchi, for example, or curry, or sauerkraut. So we have those type of preference since our childhood. We have some biasness there as well. It's not going to be really, really easy to like understand everyone's flavor goals or optimize based on everyone's flavor, but I think it is possible using Flavoromics. It is possible to develop food or drinks that certain population would like. That can be dependent on age as well, the gender as well. So we should not forget about those. So yep, it's going to take quite a while. And in New Zealand, we are a pretty small country, only 5 million people here. So we'll most probably need to work with international collaborators, where there are larger population, so that we can actually understand the flavor goals for everyone, or we can suggest something to optimize that area.
Yeah, very interesting. We have another question. It says, will cell-based assays that use human TAS2 and 1 receptors be considered flavoromics?
I would say yes. This is actually one of my personal interests as well, but we do not, like within plant and food, I don't have any collaborators to work in this space, but I'm looking for collaborators right now. And we have some idea that we would like to actually use cell-based assay to understand how the receptors are interacting with different food composition.
And also we have another question. So it says, where do you think AI and machine learning tools play a role in flavoromics? I think of molecular networking of unknowns and molecular docking. What's your opinion on this topic?
We are in the same wavelength. Yes, I think the AI and machine learning tools, we're using machine learning tools. We have not started using the AI yet, because I think AI needs to be developed a little bit more, because AI is developed in certain areas more than others. But I think this is why we most probably need to generate more data to help the AI tools as well. It would be really, really important to use AI and machine learning tools in future. And yes, molecular networking to understand and molecular docking to understand unknowns. This is quite important too. So we have colleagues in another organization. They actually use molecular docking to understand, the responses of small peptides. So yes, I think, it's really, really interesting. And to be honest, what I showed you, that I want to understand the emotional space better, emotional responses and the chemistry behind it better. I think for those, we will need to use AI and machine learning tools.
Thank you for that. We got another question from a doctoral student from India. It's asking about the details of the type of columns you use during the untargeted analysis of wine, and also about sample preparation. I think this can be seen in your papers, but I don't know if you can say something about that.
Yes, this paper is published in Food Chemistry. I provided you the reference snapshot. You will find all the details in that paper.
Okay, so we have another question. In second case study, you mentioned conceptual response and aroma description under sensory consumer panel. Can you provide some examples of these responses?
Yeah, I did provide the examples of those, like, like the aroma description, we had, like, for example, fruity, grassy, melon and coconut, those kind of descriptors, right? And yeah, in terms of emotional response, we had like exciting, bored, moorish, dull, those type of information. Is that enough, or do you want more information?
We haven't got any other question about that, but
Yeah.
we have like a related question that says that about the method, the GCMS method. Did you calculate retention peak resolutions as necessary condition to confirm a compound? I think this is confounding with other type of GC methodologies, but maybe with the paper that could be clarified.
Yep. You can find all this information in the paper.
We don't have any more questions. I think we conceptual says Dr. Nikunj Patel. I think it's about the other question.
Yeah.
I
I
have a
think so
question
too.
of my own. It's more like
Yes,
a
please.
comment. And do you think changes in the gut or oral microbiota can affect how we perceive taste or influence what we like to eat? I saw the study on the age relation, but I think more personal manner, like microbial metabolites.
So if you think about flavor perception, because flavor perception, it actually happens directly after we consume food, right? But in terms of relationship with gut, that's different topic, like it's more gut brain axis that might not be 100% related to the way we perceive. Like you eat food and it goes to your gut, the microorganisms are producing different kind of neurotransmitters that can provide you, send signal to your brain that you are feeling good or you're not feeling good. So that most probably related more to the gut microbiome, and related metabolites are related to the mood and cognition rather than like immediate flavor perception.
Yeah, I was thinking of the gut brain axis, like a reward system or something like that.
Yeah. So, but in type of flavor perception, I will say saliva or the microbiome in the mouth, they will be quite important. And the changes are quite fast. From that wine oral processing data, we have seen that it was like 30 seconds in the mouth, right? But the changes happen pretty quickly. So there were some non-volatile precursors. They just turned into volatile compounds. So yeah, it's quite fast. Microbiome has big role in everything.
Thank you.
We got like the last comment from Dr. Nikunj Patel. It says it's examples for conceptual responses.
Example of conceptual responses.
Yeah. It has to do with the sensory consumer panel.
Yeah. So I'm just looking at maybe you get in touch with me later. I can actually give you show you the ballot that we have used later on, because this is a long kind of ballot that we have used for different, and we have everything written. I do not actually recall from the top of my head right now. Is that okay?
I think that would be really helpful.
Yep.
Thank you so much. So we are going to finish the session. Thank you very much, Dr. Pinu, for your time, your availability, your predisposition, and for answering all of these questions. Thank you everyone for joining us today. We just wanted to mention that we're going to take a time from the webinar series, given that next month it's the annual MedTalk conference, and then there's the summer vacation on the Northern Hemisphere. So we're coming back in September. So just keep updated because there are going to be other events or other activities related with the MedTalk with the EMM. So please stay updated. Once again, thank you very much for joining us today, and thank you very much, Dr. Pinu.
Well, Daniela, I think I found the conceptual.
Okay.
Sorry. The emotional and conceptual responses were together, right? So emotional, like for example, the excited, enthusiastic, those kind of things. So there are other terms like on green in that plot were like traditional, balanced, good for me. So yeah, those were the conceptual, and the blue one were emotional.
Perfect.
Yeah, sorry about that, because as I said, sensory is something that I am still trying to understand, and I don't think I can, because I was thinking that I could call Emma and ask her to explain it, but I think it's not like, you most probably muted everyone in the attendees. So yeah, no, that's why. But yeah, if you want to get more information, please watch the space until we publish, or you can get in touch with me.
Thank you so much. Thank you, everyone, again.
Thank
Bye.
you. Thank you. Take care. Bye.
Bye-bye.